Hey everybody, and welcome to chapter number 2. We are talking about the eye today. This is kind of a tricky chapter because it really does feel a lot like anatomy. It doesn't really feel like psychology very much in this chapter, so just a fair warning. Also, those of you that have taken a class in anatomy, I know that some of you for your core, you may have taken human anatomy and physiology. I've had a lot of students take this class and say like, hey, I'm so glad we covered chapter 2 because we learned about that in my other class. So it is going to be a little bit heavy on anatomy, but that is okay. It is a little bit of a shorter chapter for that reason, because we're talking less about processes and more about structure. And honestly, for the remaining chapters, or the remaining senses, I should say, we'll generally be spending time talking about the anatomy of that sensory organ, and then how that anatomy turns that turns that stimulus into something that our body can detect and something that our brain can understand and make sense of. So let's get into it. We're talking about the anatomy of the eye. So we're going to be talking very briefly about the properties of light. We'll talk a lot about the structure of the eye, and then we'll kind of conclude by talking about photoreception, which is also known as transduction. Generally speaking, anytime we talk about the point where the physical becomes the subjective, or becomes the internal, becomes the personal, becomes the perception, we call that transduction. So your eye turning physical light into something that your brain can understand, which is just action potentials. If you remember that from last chapter, that process is called transduction. We got it for our eye, for our ear, our tongue, our nose, and so on and so forth. Let's get into it. I have a couple of videos for you here today. Excuse me. I'm going to show you one real quick. These others, I think they're interesting, but they're a little bit dated. And actually, all of these are dated, and I'll mention why in just a second. So I'm going to tab over where we're going to check out this lab. Here we go.
Hi Recon, welcome to our laboratory. Today we're going to show you how the transcranial magnetic stimulator works. So first thing, Yoshi's going to put the magnetic coil over top of the area of my motor cortex that's responsible for the muscles of my hand and forearm. Once she's there, she's going to deliver a magnetic pulse. Oh, and you probably saw my arm twitch there. And we can also see this response over here on the oscilloscope, because we've measured it using the EMG electrodes over the muscles of my hand and forearm. Now what we're going to do is move that magnetic coil over the span of my motor cortex from one side all the way over to the next. And what you'll see is muscles slowly recruited from my hand up to my elbow, shoulder, right leg. And after she switches hemispheres, we'll see some of the left leg come in, and then down the muscles of my left arm. So here we go.
Okay, now before this exercise continues, I want to mention a couple of things. This comes from chapter 1. You may remember we talked about there are different neuroscience techniques that researchers use to understand the connection between the physical external world and the internal world, specifically how the brain operates and how the brain works. And what we are seeing here is TMS, transcranial magnetic stimulation. And what this lab assistant is doing is she is placing that really strong magnetic pulse above different parts of her cortex. Now, you can kind of see that it's around the midway part of her skull. It's not super close up front. It's not really in the back. It's right there in the middle. Because as we're going to talk about in chapter 13, our bodily control, the surface of our skin, the area of the brain that translates those signals that occur on the surface of our skin are exactly where she is pointing that coil. And so we don't have to go, we don't have to, and this is a lesion study where we're looking at how the loss of brain function in one area, how that impacts the rest of the body. Now, we don't need to carve out part of her brain to understand that. A lot of the research in this area, a lot of the early research, like I mentioned with Phineas Gage, it's people that are getting injured and then we're seeing how it affects their behavior. Here, this is a temporary solution where we're able to temporarily deactivate that part of her cortex for just a fraction of a second and see how it impacts her behavior. So what you're going to do, or what you'll see here, is as she moves this coil across the surface of her head, you're going to see how at first this hand is moving, and as she moves it further, that the rest of her arm is going to start moving, and then once she crosses over hemispheres into the right hemisphere of this participant, then we're going to see her other arm. That's a conversation that we're going to be talking about a little bit later when we talk about what they call cross-lateral hemisphere activation. Cross-lateral because activation in one side of the brain is affecting the other side of the body. But we'll talk more about that when we get to that next chapter. Here we go. Here's the rest of the video.
So right hand, little bit of elbow, some shoulder and leg, still right leg, little response in my left leg there. We've switched hemispheres. More leg on the left side, left shoulder, left elbow, and left hand. Thanks for joining us.
Okay, how nice, right? So, and also you can tell this video is dated, not just because the image resolution is low, but look at those Dell computers. I feel like that really has some sense memories attached to that. I remember those growing up, those old Dell computers. Maybe you remember them too. And the USB were right up here, and they were always, you never ever put it in the right side. You always put it in once, and it's always the wrong side. You have to flip it and then try it again. You remember what it's like. Anyways, so I mentioned right before I started this video that one of the reasons why this is a little bit dated is because the newer videos of TMS are a slightly different angle, and that's because TMS has been used a lot when we talk about treating mostly depression, medicine or pharmaceutical resistant depression. And so if you Google this stuff, you can even see here, over here in the second time getting TMS before and after. It's got a couple of different recommended videos about that. So if you see something about TMS curing depression, that's not just some pseudoscience. That is something new that people are using a lot these days. Really interesting. Outside of the scope of this class, though, I would love to talk more about it, but I know you guys got to be home soon, so I want to cover the rest of this stuff. So these other videos, they're interesting, but not as important as this one. So if you're interested, check those out. All right, so let's talk a little bit about light and how it works. Some of you may know that we don't really see most light around us. When we talk about light and light waves, those include television waves, those include radio waves, those include microwaves and heat rays. And you probably have some conspiracy theorist in your life or on social media who's always talking about how, like, the Wi-Fi signal, it's scrambling our brains and stuff like that. And it's like, yeah, we can't see Wi-Fi signal because Wi-Fi signal is imperceptible visually. It is not something that we can see. We can only see a very small band of light, and that band runs from 700 nanometers to 400 nanometers. But within that 300 nanometer interval, we can see all the colors from red over here down to blue. And yes, it is the colors of the rainbow. So we're moving from red to orange to yellow to blue to indigo to violet. You remember Roy G. Biv? Oh, I forgot the G over here. But yeah, so the rainbow, that encompasses the extremes that we can see in terms of color. We'll talk a lot about color later in the semester in Chapter 5, but for now, even if you weren't able to see color, you'd still be able to see light if it is 700 nanometers. You'd still be able to see it if it's 400 nanometers. But if light is at 710 nanometers, you cannot see it. No matter how hard you try, you can't see it. It's imperceptible to the eye. Not imperceptible to all earthly creatures, though. As some of you probably know, there are animals that can see ultraviolet waves. And I think certain species of shrimp can see an infrared, I believe, or that one also might be ultraviolet. Either way, one fun fact here is that crows and other corvids like ravens, they appear to our human eye as kind of solid black, right? You think about a crow, it's mostly, a black bird. But to a crow's perception, crows actually have very colorful plumage to their own eyes. They're actually able to see a little bit further down here. They're able to see in the ultraviolet, and so their plumage is black and whatever that ultraviolet color would be to our eye. So they actually are sparkling and dazzling and look really, really cool. We see them as kind of, this, just kind of unicolor animal. So that is what we can see. So everything we're going to be talking about in the next 8 chapters is going to be about this small band of reality that we can perceive. There's so much more reality outside of us that we cannot perceive, though, which is really, really interesting. Hey, who knows? Maybe the Martians that are also taking this class that can see microwaves, maybe they're just talking about this band of light right here and they can't see this. They can still see visually, but they would not be able to see any of the things on the computer screen in front of you, for example. Anyways, really fun thought exercise. Something to think about when you're in your dorm room late at night. But for now, let's keep moving. So we have a lot of chapters about vision. I don't know if you saw that in the textbook. The textbook, has 15 chapters, and of those 15 chapters, let's see, 1, 2, 3, 4, 5, 6, 7, 8. 8 of those chapters are dedicated to vision. It's not until chapter 9 that we talk about hearing. So why is it that literally more than half of our textbook is about vision? I would love to ask you directly instead of waiting for you to waiting for this video to continue. I would love to interact with you in a classroom and ask this, because I usually get a lot of interesting answers here where people will say there are a lot of chapters about vision because vision is very complicated. Okay, maybe it's because vision is really important to everyday life, which is also true. I think actually the answer is in some ways more simple than that, but also a little bit more annoying, which is that's just where the grant money is. That's where the money to do research, a lot of that stuff goes towards visual things and less towards hearing. And yes, that might be because we use vision a little bit more in our everyday lives than hearing. But that's part of the reason why our textbook covers vision so much. There is a lot to know. We know a lot about vision. Of our 5 senses is probably, well, actually it is the most studied. I was going to say maybe it's the most understood. I think maybe our sense of vestibulation might be the most understood. That's a really fun argument that we can have a little bit later, though. So there's a couple of reasons why we are going to be spending half the semester talking about vision. One is because vision is really complex. It is really complicated. It is not like an easy process that happens in the human body. It is a very advanced biological system. Also, we do use it more than other senses, generally speaking. You can do a thought exercise of which of the following would disrupt your day-to-day life more, starting right now. If you lost all vision, or if you lost all hearing, or all taste, or all smell. I didn't say touch because that, depending on your follow-up questions, might be very interesting. And we'll actually look at people that are born without the sense of touch. But most people in this thought experiment will say that they would rather lose their sense of smell or their sense of taste before they lose their sense of hearing, and all of those before they lose their sense of vision. And that's because as a society, our day-to-day lives are very visual based. We think about if I were to ask you to come to Westfield's campus, could you do that without being able to hear? Yeah, probably. Could you do that without being able to taste or smell? Yes, certainly. Could you do it without being able to see? That's a lot more complicated. So we use it for navigation. We also use it to determine what foods are something that we want to eat and what things we want to avoid. You can think about looking at rotten food versus good prepared food. mate selection, which is a fancy way of saying dating, right? That when nobody likes to admit it, but we do care about what people look like and how they present themselves visually. We rely on vision for safety because we don't want to run into a wall. We don't want to drive off the road. We don't want to fall off a sidewalk or a bridge or whatever. We rely on signs to help us get around. And hey, what are you doing right now? You're watching a lecture video. If we relied more on hearing, that might make my job a little bit easier because I could just record these as a podcast, right? Where I wouldn't think that you would need some kind of visual element here. But for reading, for communication, a lot of that is done visually, and yeah, pretty much everything that we do has some kind of visual component to it. I just said podcasts, which is interesting because some of you may know in the early days of podcasting, it was just audio files, right? People just would upload these MP3s you can put on your iPod or whatever. But now, in the past decade, what we're seeing is that people like having some kind of visual component to that. When people watch Joe Rogan, or when they listen to Joe Rogan, for example, they might prefer the video version of that podcast. So even the audio stuff has visual components to it. But I've also had students in this class take this class who are non-sighted, who are not able to see, and I'm sure they could probably tell you that their lives are very full and very complex and meaningful without vision. It's just that our society is just very vision focused. So let's get to know this sense a little bit more intimately. Let's talk about the structure of the eye. So here it is. Here's the structure of the eye. Better take a picture, but we're going to walk through kind of what a lot of these key pieces of anatomy do, and what they're for. But here is essentially the way that we're looking at this is that light is going to enter moving from left to right. It's going to enter in through our cornea. It's going to pass through the aqueous humor, which is this area right here. It has a lot of fluid. It's like a pocket. It has a lot of fluid in it. From there is going to enter into our pupil, which is what you're seeing me draw right here. Our iris, which is what gives us the color of our eyes, not the white parts, but the maybe it's brown, maybe it's blue, whatever. Maybe it's hazel. That is going to be this muscle right here. We got our lens. This is what's going to dictate if we need glasses or contacts. Is this lens right here and how good a job it does. For most of you. And then we have the retina back here, and the retina is where transduction happens. And then once we talk about that, then we're pretty much done with the chapter. So let's get right into it. Let's talk about this stuff. So here is a cross section, and again, if you want to kind of follow along, we're looking at light moving downwards now. So light is ultimately going to pass through all of this and hit the back of our eye, just like that. Okay, so we're going to be talking about the cornea first. So this cornea, the cornea is the transparent window from which light passes through so that it can get to the rest of your eye. The reason why it is transparent is because it has no blood vessels. That's how it's able to be transparent, which also makes it quite sensitive and easy to damage in a long-term way. Because if you were to scratch your cornea, it takes a long time for that part of your body to heal itself because you do not have the advantage of blood vessels to help provide nutrients. And so if you're like, but wait a second, how the hell does that work? If there's no blood there, how does it get nutrients? How does it get the enzymes needed to keep those cells alive? That is because of this area right behind it, the aqueous humor. So this pocket right behind the cornea, this area is going to be what supplies the cornea and other parts of the eye with with oxygen, and that oxygen is going to keep those cells alive. It is also going to remove waste that gets trapped back there. So that's the aqueous humor. So light is coming in, is passing through this transparent window, and it's going to keep moving through this fluid. All right. From there, we have the iris, and as I mentioned, the iris is this piece right here. It is the part of your eye that might give your eye its quote-unquote color. So, I'm going to get right up on the webcam for a second. But, like, if you are looking at an eye, you'll note that I have kind of, like, bluish-gray eyes. That is because my iris is a muscle right here, and the pigment in it is bluish-gray, and that is genetically determined. And you probably learned about this when you're learning about Punnett squares and all that in middle school, and about how you have recessive and dominant genes, and that those can dictate what color eyes you are likely to have whenever you were born. So that is from this muscle right here. It is a muscle. We talk about certain parts of our eyes constricting or expanding. That is going to be the iris doing a lot of that work, but we'll talk about that in a second. So the iris is a muscle, and that muscle is going to tell us how much light we let into our eye. So if we have light coming in, you can see that this pocket right here that I'm highlighting, I'm just going to color it red. This is how much light can pass through. So all light that goes in through here. Now it's up to the iris to determine if we let in more or let in less. As some of you probably know, whenever you are in low luminance settings, if you have the lights turned down really low or your night vision has kicked in, that the pupil is much thicker, that it has a bigger diameter. It's letting in more available light. However, if you're staring at the sun, much like Gustav Fechner did in Chapter 1 and almost blinded him, your eye is like, whoa, we don't need that much light. Are you crazy? Do you want to be like Gustav Fechner? So it's going to, your iris is going to try to constrict and try to let in as little light as it can. So whenever light is bright around us, we don't need as much, so our pupils get really small because this iris is flexing, essentially. But I use the word pupil, so what is a pupil? A pupil is an interested student. That's hilarious, right? But it's not hilarious, and I'll tell you why it's not hilarious, is because we all know that pupil is another word for an interested student, but no one ever uses that word, right? Like, has anyone ever used that in a common everyday way? I teach for a living, and I can't tell you that the last time I've ever used pupil to talk about an interested student unless I was making this joke. So the pupil is that the part right dead center in your eye that is black. It looks like a hole because it is a hole. What a lot of people don't realize about the pupil before they take this class is that the pupil isn't necessarily a structure. It is a window. It is, fancy SAT word here, it is an aperture. It is an opening that is going to allow light to come in. But it's not actually like, we don't have like a black dot here. It's black because it's really dark in our eye. So light is going to enter in through that hole whenever we are in low illuminance settings, when there's not a whole lot of light out. Then that pupil is going to get larger because our irises are relaxing. That those muscles are relaxing and letting in more light. Whenever we are in a bright setting, it is going to constrict and it's going to let in less light. All right. Now we are moving to our lens. The lens is back here, and I mentioned that this is the area that is responsible or guilty for whether or not you need contacts or glasses. So we got light coming in through our cornea. It's cutting in through the fluid in the aqueous humor, and then it is reaching this window in the pupil, and then from there it's going to hit the lens, and the lens is a lot like a lens, like a photo lens or a magnifying lens. It is like a piece of glass. It's not really a piece of glass, but it has the same kind of idea where, like, if you have some glass and you try to, like, okay, I'm going to give you an example. I don't know how well this works for you because I don't know how bored of a kid you were, but I remember seeing on TV or in the movies where kids would have magnifying glasses and they would try to burn ants. with that, using the sun's light, right? You've probably seen that before. I don't know if you've ever tried it or not, or if you ever tried to light something on fire using a magnifying glass. But if you were doing that, you have to hold the magnifying glass at just the right angle. If you don't, then the sun's rays don't they don't refract on the right spot. It doesn't refract at a pinpoint. It doesn't react. It doesn't kind of come together in one point. Maybe if you turn the lens a little bit, then it is kind of blurry. There's less light and it's more scattered and it's not kind of a pinpoint. That's what we need from our lens. Whenever we have light coming in through this, we need that lens to essentially kind of focus that light that's coming in. So you have all this light coming in from around us, right? So it's all coming in, and we need it to really kind of, especially the stuff right in front of us, to kind of refract at a perfect point down here in our retina. But some of you, if you need contacts or you need glasses, it's because that image doesn't refract perfectly right here on the retina. Instead, it might refract a little bit behind that. So it's kind of like refracting perfectly back here. Whoops. Actually, let me just erase all this. So it's not perfectly on the retina. It's from, looks like right here. Yeah, that's much better. Which means, as you can see right here, that now kind of this much of the retina is kind of being occupied by this visual image instead of that little pinpoint, which is going to cause your vision to be very, very blurry. Or we can have essentially the opposite happening, where instead of focusing on a pinpoint here in the retina, it's focusing a little bit short of it, which means that it's then going to unrefract like that, and then now again it's blurry again. So whenever we need glasses, it's almost always because of our lens. It's going to help us focus that light coming in to our eye. Accommodation is the process in which you can kind of focus or unfocus your lens. Your lens, its shape is controlled by these muscles right behind your iris. These are called the ciliary muscles. The ciliary muscles. And these ciliary muscles are why, for some of you, for example, whenever you're like reading, you kind of got to squint a little bit, right? Well, I don't know. Some scientists who are actually watching this might not like that example, because there's actually multiple explanations for why whenever you squint, things can be a little bit easier to read if they're close to you. But for the sake of argument, whenever you are trying to change your focus without changing where your eyes are looking, that is because of the ciliary muscles are either gently pushing up against the lens to make it fatter or to make it thinner and more narrow. Either way is going to have an impact on where that light is refracting. That is called accommodation. Okay, so let's focus. You see, got another joke for you. Let's focus. Now, the reason why that's funny is because I'm talking about the lens, and the lens is helps us focus the refractional power of light, but the humor comes from the other meaning of the word focus, which means to allocate your attentional resources to a specific thing. All right, I knew it wasn't funny, and I thought that maybe explaining it might make it a little bit more funny. Sorry about that. So when we talk about, as I was mentioning, when we talk about the lens, there are problems that can arise because of a misshapen lens, or a lens that is too narrow or too wide. So the two big ones here are myopia and hyperopia. Myopia is known as nearsightedness, and myopia is whenever light is focusing in front of the retina. So just to back up right here, that's what we see right here, that light is focusing to a point before it gets to the retina. That's going to lead to myopia, which means that whenever you're reading something, as long as it's close to you, it's okay. It's just that the distant stuff is the stuff that you have a hard time seeing. This is probably the most common, and it is usually the one that as you get older, you're more likely to get. Hyperopia, on the other hand, is where light is going to be focused behind the retina. So it's the first problem with vision that I was telling you about in that previous slide. So stuff that is close to you is really, really blurry. The stuff that is further away from you, you can see, so it is farsightedness. And then we have astigmatism. And so I kept saying, like, this is true for most problems with vision. Astigmatism is different than the other two. It's different from nearsightedness and farsightedness because it's way more expensive. And maybe one or two of you that have astigmatism that are watching right now, maybe you let out a sigh of I don't know, maybe you almost laughed because you're like, yeah, it is. What is with astigmatism being so much more expensive? The reason why is because astigmatism makes your eye unique. That generally with myopia or hyperopia, you can get a prescription, and that prescription is like, you want to have plus 0.5 refractive power to this lens, and you want to have 0.25 in the other lens or whatever. And then, hey, 1 second, y'all. Alright, sorry, I had to pause it because as you can see in my camera right here, this little guy right here, Fozzie, he was trying to get into my office door and I had it closed, and he was looking very pitiful, and so my spouse was like, you need to let Fozzie inside. I think he's going to be a good boy. He's going to go to sleep. Alright, sorry about that, y'all. We're talking about astigmatism, and the thing about astigmatism is that it makes your eye unique because if you had that prescription I was telling you about earlier, and your friend has it, then you can swap glasses pretty much, and it's totally fine. But if you have astigmatism, your prescription are kind of going to be very particular because it's not going to be just, hey, this one eye needs to zoom in. Instead, it's like the left portion of this eye needs to be zoomed in, but the right portion is fine. And then for the other eye, it's the right portion needs to be zoomed out, but the other portion of the eye, the left portion of the eye, needs to be zoomed in. Basically, it is an uneven distribution of light because the lens is not perfectly shaped. It means that it's a little bit lumpy, and because of that, that light is not getting perfectly on the retina. So here is emetropia, what we call quote-unquote corrected or normal vision, where we have that light is refracting perfectly right there on the retina. Myopia, here you can see where it is refracting a little bit early. And then hyperopia, which is refracting a little bit late. And so if you have myopia, the way to correct for this, the way that contacts or glasses work, is because you have this light coming in, you put something in front of the lens, and maybe that's a contact, maybe it's your bifocals or whatever, but basically you can see that light is starting to refract a little bit in the other direction now. It's making that image a little bit bigger, and then by the time it gets to your cornea, then it's going to behave like normal, and so it actually hits it at just the right spot, because now this image is slightly bigger than it is over here, and that is going to help it refract perfectly on your retina. Here is an astigmatism test. So if you think you might have astigmatism, just check out where you see the cursor right here in the bottom. And if you're staring at that and some of those lines appear to be wiggly or curved in some way, then you might have astigmatism. So you can close one eye and just do it with one eye at a time, and that can tell you, like, oh, the lines on the left are bad, or the lines up top are bad, or the lines over on the right are bad. But either way, essentially, this lens right here, it could be where instead of being this perfect oval right here, maybe there's a little bit of a bump right here, and that's causing that light to not reflect perfectly or refract perfectly on the retina. Okay, I've been talking about the retina a lot, and again, if you are worried about your vision, I would be happy to answer any questions you have, but I am not an optometrist and I'm not an optician. I don't know how much I can help you other than to recommend you check out an optometrist. All right. So, in terms of the retina, I don't need I don't have glasses right now, but I probably do need them. My dad was about this age whenever he started wearing glasses, so that can happen too. That over time, your lens is no longer going to refract perfectly back here. All right, so the retina. we've been talking about the retina a lot. What exactly is the retina? The retina is made up of rods and cones, and together that makes the retina something that is light sensitive. In other words, these are cells that are going to respond to the presence of light. That makes them essentially unique in the human body. I had a student one year when I was like, these light sensitive cells, we call them photoreceptors, and they are the only cells that can that react differently when exposed to light. And I had a student raise their hand and say, does that mean that a chameleon's body is covered in photoreceptors? And I said, no, but I have to get back to you on that. And that student was right, that they're not actually photoreceptors on the surface of a chameleon, but they are cells that are sensitive to light. So if you have camouflage skin and you can melt into your surroundings with camouflage, then maybe these are not the only light sensitive cells in your body. But for all of us, except for you, these are the only cells in our body that really kind of change their activation depending on if there's light present or not. If I cut open your skull, pulled it back, and shined a flashlight onto the surface of your brain, you wouldn't feel that, you wouldn't see that, your brain would not react to it. It does not have the hardware to detect that light right on the cortex of the brain. It's only these parts of our eye, those photoreceptors. They receive light and they turn it into a neural signal. We call that process transduction. So transduction is when these cells are going to convert the presence of that light into an action potential, into a neural signal that can then be sent down these different pathways and make it to the occipital lobe and the brain. So those neural signals, if we see something over here because we saw something right there, that signal is then going to come all the way down, leave out the back of your eye, and then make its way to the back of your head, to the brain. The retina is lined with photoreceptors, and we call those rods and cones. We'll talk about them a little bit more in a second. I want to spend a little bit of time here talking about age-related macular degeneration. This is a form of vision loss that generally happens because of old age, and that as we get older, that the central parts of our visual field are going to be degraded, and this is again another very common problem that we have in sensation and perception, is that we have different parts of anatomy that are referred to different things by different fields. So I talk about the retina and I call it the retina, and if you come back here you can see this F word, the fovea. The fovea is the center part of your retina. I call it the fovea because my background is in, neuroscience, cognitive psychology, neuropsychology, whatever you want to call it. If my background was in medicine, if my background was actually, if I was a physician, I'd probably call this area the macula. They mean the same thing. It's just from different areas. One's medicine, one's neuroscience. The macula is the center part of your retina, and as we get older, some of those cells can die off, and that presents an issue with being able to detect the light directly in front of you, and this is called a scotoma. So in this image, you can kind of see back here, it is pretty blurry on the whole, but it's most blurry right here in the center because of a scotoma, where they are not able to see in the center part of their visual field, which means that the periphery is still generally okay, but it does get worse and worse with time. If you want to check out what that looks like, you can check out this link right here in your lecture slide notes. Okay, so I mentioned that that those cells, that are creating an action potential, they're creating these neural signals, they're leaving in the back of the eye right here, and that is called the optic nerve. This optic nerve is a bundle of these axons from those cells that are going to try to communicate that presence of light to the back of the head. And each eye has essentially this little hole right here. We call this the optic disc or the blind spot. The blind spot is a spot in our eye that we can't actually see anything. We literally cannot perceive or cannot detect any light here because there are no photoreceptors there. That's where those axons are leaving the eye so they can get to the rest of the brain. But if you're thinking critically here, you may be thinking like, but wait a second, what do you mean a blind spot in our eye? And then maybe if you think about this, you'll think like, okay, well, if we have two eyes, then that makes sense. You know, my one eye over here is going to cover for this one, and this eye is going to cover for the blind spot in that. But then if you close one eye, it doesn't really change anything, right? There's not a blind spot now that you close one eye. So what's going on with this blind spot? This is a really great example of the difference between sensation and perception. Because if we're talking about just straight up sensation, which is the detection of physical stimulus, we're not actually detecting any light whatsoever. We can't. We don't have any rods or cones or any photoreceptors here to detect that light. But we still perceive something to be in front of our face. We don't perceive there to be like a big hole from where we're not where we're not detecting anything. So we're perceiving light, but we're not actually detecting it. So what's going on here? You can try to find your own blind spot at home. This is another one of those cases where I hope that you take time to pause this video and to try this out, or you can even Google on YouTube, how to find your blind spot, and I'm sure you can find some cool exercises for this. But here's how I recommend doing it in the classroom. That basically you want to use one eye, so I'm going to cover up my left eye, and so I can only see with my right eye right now, right? So I want to try to find the spot in my eye where I don't have any photoreceptors. The easiest way to do that, where you have one eye covered, is you give a thumbs up like this, and you keep your arm roughly perpendicular to your body. So it's at a right angle. It's just sticking out straight ahead, and you're giving yourself the thumbs up like this. And if you have it right here and you're looking straight ahead, so do not look at your thumb. You're looking straight ahead. There will be a point right around here where you can't really make out your thumbnail, where it becomes kind of blurry, where you can't really see any details. And of course, if you move your eye to look at your thumb, then yeah, you can see your thumbnail, you can see the wrinkles in your thumb or whatever, it's totally fine. But as long as you're looking straight forward, you lose that definition, that it looks really, really blurry. That's your blind spot. And that's a really cool example of how you're not actually detecting your thumb, but your brain is essentially saying, I think the most likely thing to be here is this. And it creates something there. Something that's not very vivid, something that's not super detailed, because it doesn't want to get our attention, because if we were constantly looking at where our blind spot was, we would be looking all over, right? So anyways, kind of an interesting thing, right? And again, if it doesn't work for you, try it again. Try it while not looking in this direction. Try finding another video on YouTube where you can try to find your blind spot. It's definitely like a cool, fun thing to do. All right. Anyways, so let's talk about photoreceptors. So those photoreceptors, those rods and cones, you may have heard of rods and cones before. They are two different types of photoreceptors. Generally speaking, the rods are going to be used for black and white, and the cones are used for color. A nice way to remember this: cones, color, C-O-C-O, cones, color, color, cones, color, color, cones. The cones are going to be what dictate our ability to perceive color, and if we are color blind, again, we'll talk more about that in Chapter 5. Excuse me. And rods are going to be used for a lot of peripheral vision, I will say. Okay, so rods don't process color. They're used in low luminance conditions. In other words, whenever there's not a lot of available light, we're going to rely a lot on our rods. So we're using them mostly for night vision. And they have low visual acuity. Visual acuity is a concept we're going to be talking about in the next few chapters. One way to think about the word acuity is to think about definition, if you're familiar with like HD, like high definition versus standard definition, low visual acuity is essentially like saying like standard definition, where the pixels, right? So think about like, and actually if you want to, you can take this video and you can crank it down to like 320 p, which would make it kind of blurry, or you can crank it up to 1080p, you know, so that it's high definition, and we would say that that's high spatial acuity. What we mean by that is with low spatial acuity that the pixels, the individual blocks that make up the screen, are bigger. And because they're bigger, we can't get a lot of really small details in the visual image that we're trying to get. And so if you want something to look realistic, you usually want it to be in high def, so you can see all the wrinkles and all the freckles and all the hairs or whatever on that person's face, and it means that the little blocks that make up that video are much smaller, and so they're closer together, and so you can represent detail and specifics much more easily. So that's all just to say that think of rods as being standard definition, and think of cones as being 4K. That it's like super high definition versus pretty low definition. That's what we mean by acuity. Cones can process color. We'll talk more about that in chapter 5 though. Here's a table you can also find in your book that just walks through this. The only thing I want to highlight here is that the book will also talk about the cones as coming from the photopic system and the rods as belonging to the scotopic system. So photopic system is kind of like saying your color vision. Scotopic system is kind of like saying the black and white peripheral vision that we have. The other thing I want to point out here, and I got another figure to kind of illustrate this, but in terms of where things are in our retina, that most of our cones are in the center of our eye, close to the fovea, or the macula, for those of you surgeons and physicians watching this video. And our rods are mostly going to be in the periphery. They're mostly in the outside part of our eye. Each rod is going to be powered by a protein called an opsin. An opsin is just going to be essentially like the engine that powers that specific cell. For our rods at least, we only have one type of that, and it's called rhodopsin. The interesting thing about rhodopsin is that whenever it's dark, if we have our night vision, our rhodopsin is totally fine, it's working great, and it's powering the cells that allow the rods to do their job. If you turn up the light, if you have all the lights turned on, if someone shines a flashlight in your eye, if you open up the refrigerator and it blinds you for a brief moment, what's happening is that rhodopsin, the pigment that makes, the protein that makes that cell powered, that keeps it online, is disintegrated. It is decombined, and so that cell, that rod, can't really do its job. That's why whenever it's really dark outside and somebody shines a bright light in your face while you lose your night vision, is because that rhodopsin has now been broken down because of the bright light, and it takes about 30 minutes, 25 minutes for that rhodopsin to work again, for it to slowly kind of recombine and power that cell again. Cones are a little bit more complicated because they can fall into one of 3 different types of cones: a blue cone, a green cone, and a red cone. And we'll talk more about that in color vision, because depending on what kind of wavelengths those cones see, if they see the short wavelengths, the medium wavelengths, or the long wavelengths, that can actually tell us what colors they are able to perceive or detect, I should say. Here is an ink staining of someone's fovea. Here it's interesting. There's a lot of these red cones you can see, a lot of green, fewer blues, and it's not really until the periphery that we can kind of see the rods, right? So in the center here, it's almost exclusively cones, and one of my favorite figures in the book, you can see it right here. Here, the way to interpret this, on the x-axis, we have how far away is it from our fovea, and with 0 being the dead center of our visual field. Way over here on the left, we are really far on the left part of our visual field. Over here on the right, on the really far end of our right visual field. And we're talking about our unit of measurement is in microns here. This right here is in microns. And this down here, the x-axis, is in millimeters. My bad. On the y-axis, we have the density of those photoreceptors. So we have higher density up top, and we have lower density down the bottom. And so, looking at the cones, for example, let's trace the red here. We have essentially 0 cones until we get right up on the fovea, and then it shoots up. We got a lot of cones right here in the center part of our visual field, and that's pretty much only where they are. And if we look at the rods and trace that over here, we got some rods in our periphery all the way to the left. We're getting more and more rods as we get closer to the fovea, but then once we start getting right up on the center of our visual field, it starts to fall away because we only have cones right there. And then we have the same thing happening but in reverse for the right part of our visual field. I love that the figure takes time to say, here's the optic disc. In other words, there is no rod, there is no cones, because this is the part of our eye where those axons are leaving that anatomy. I already mentioned all of this about our eyes adjusting to the dark, but that light can disrupt our ability to see. Over time, that rhodopsin can recombine itself back to its kind of base rate, and then you're able to see. This can take about 25 minutes for you to be able to do that. And the other thing that happens along with that, so whenever you are adapting to night vision, there are kind of two things that are happening. One is this. One is that our rods are kind of preparing themselves to go back online and to be useful again. So the rhodopsin, now that there's not a lot of high intensity light around, bright light, it has a chance to kind of rebuild. And the other thing that happens is that our pupils are going to get wider. When our pupils get wider, it's letting in more light so that our rods can get all the very low levels of light that exist. Maybe it's from the stars in the sky, maybe it's from the night light in your bedroom, but your eyes are able to detect that because your pupils are bigger, they're larger, the irises have relaxed, and so more light is able to get into your eye. Here's an example where we got Gustav Fechner's eye right here. This MFer's looking, he's staring into the sun. And then we got somebody who just got back from the optometrist. Their eyes are extremely dilated. That's why if you've ever gotten an eye test and you have to wear those sunglasses, this is the reason why. It's because they dilate your pupils so they can really see back there. Make sure you don't have astigmatism or whatever, but they can see back there a little bit more easily whenever it's dilated. But that also means that way more light is able to get into your eye than usual, and which is why you have to have sunglasses. Retinitis pigmentosa is also another form of vision loss, and this is hereditary. This is something that can actually be passed down from your parents and grandparents. And retinitis pigmentosa is where your peripheral vision is affected first. So it's almost the opposite of age-related macular degeneration. For retinitis pigmentosa, it's going to kind of eat away your periphery first, so you start to lose all your peripheral vision, so that you're mostly just left with the center of your visual field, the high def colorful part of it, and then over time that will go away too. It gets smaller and smaller and smaller until you can't see anything anymore, and you can see a lot of the cell death going on right here. A lot of that taking place. With that being said, this is one of those things that is maybe on the horizon to change in the next 10-15 years, where people have looked at how stem cells can be used to actually rejuvenate photoreceptor health, but also, as we're going to see in the hearing chapter, inner ear hair cells, which are great for hearing sensitivity. Okay, this part of the book I'm going to kind of breeze over because I feel like it is way more detailed and in-depth than what you would need for the exam, but really for the real world. The book talks a lot about the different kind of layers and slices of the visual system, specifically the retina, where we have ganglion cells, we got horizontal cells, bipolar cells, and amacrine cells. The way to interpret what you're seeing here, we have the retina and the photoreceptors in the back over here. And so light, this is how vision works. Light is going to come in this way, and it's going to go through all of this tissue. So that light, it just goes straight through this tissue, and then it's detected by these photoreceptors back here, and then from there, then it works in reverse, and it sends that signal over this way. So it sends it this way, then it kind of synapses onto horizon cells, which can kind of spread out that visual information, or bipolar cells, which connect with the ganglion cells, which are the axons that are going to travel outside of the optic disc. Don't worry about the details on this. I would say the most important thing to take note of here is just that light is passing through this tissue, and it hits the very, very, very back of your eye before kind of working its way back up as neural energy. So it's transduced right here in the very, very back. It turns into an action potential, and then that signal is then sent forward through this tiny slice of the retina so that it can get out of your eye. Visual angle, we're gonna come back to this a little bit later. I'll talk about that in the next video, because we'll be talking about visual acuity and spatial acuity a lot, which would be a good place to talk about that. What I do want to conclude with, though, is introduction to receptive fields. So receptive fields are going to be ganglion cells and how they are sensitive to where light is activating throughout the retina. The reason I'm being careful here is because ganglion cells themselves don't actually detect light. The photoreceptors do. And when those photoreceptors are sending their signals, those signals are hitting other cells, they're hitting these ganglion cells, and depending on where they hit in those ganglion cells, that tells us where that light is. Does that make sense? So the ganglion cells are going to tell us essentially where is the light. So photoreceptors are like, is there light? Yes or no? And then what's going to help us kind of figure out where that light is going to be the ganglion cells. So we have two different types of ganglion cells. We have on-off cells, which are sometimes called on-center cells, and we also have off-center cells, or off-on cells. And the way these work, I'm going to actually move to this slide right here because it does a better job of being able to see it, helps communicate it. That's the whole reason why we have more than, we have 8 chapters of vision, is because it's much easier to understand these things when we see it. So this is an on-off cell, an on-center cell, also known as an on-off cell. And so this cell right here, if it's getting activation from cells saying like, hey, light is right here, you can see how that changes this particular cell's response. It goes from kind of this baseline where it's just kind of naturally kind of firing a signal, every now and again, just naturally on its own. And then whenever light is presented right here, now it's firing a lot. And then you turn the light off, and then it goes back to normal. Bop, bop, bop. So if we do that, basically what the cell is saying is like, wait a second, there's a lot of light happening in the center part of my field. There's a lot of light happening right here in the center part of that cell. If we look at that same amount of light and we move it, let's say right here to the surrounding part of that cell, look at what happens. It's the opposite, where we go from baseline again, the natural kind of occurrence of action potentials. Sorry, that was hard to read. Let me try that again. Baseline. So baseline, the natural cell responses, and then whenever light is now projected onto the surrounding part, now that cell shuts up It gets really, really quiet. And you may remember back in chapter 1, I said that whenever we talk about how the brain communicates with itself, that it knows 3 words. More, which is what we're seeing here. Less, which is what we're seeing here. And then the same, which is what we're seeing in the baseline areas right here. Over here, you can see that whenever this light goes away, we have kind of a compensatory inhibitory response. We'll talk about that a little bit later. But this is part of why we are able to see after images for motion, for color, and things like that. But we'll talk about those in chapter 5 in a little bit more detail. But for now, what I wanted you to focus on is that for these ganglion cells, if this cell, let's say that light covers the entire part of that cell, the center part, I should say, that this cell is going to be at its most excited. This would cause this cell to fire as fast as it possibly could. If you take light and shine only the periphery, then this cell would be at its most quiet. It would not be firing at all. It would inhibit all of those responses. So again, what these cells are communicating is not just is there light, but how much light is actually occupying these different areas. If this cell right here fires its fastest, then we know that means that light is only right here in the center part of that cell. If that cell is extremely quiet, the quietest it's ever been, then we know that light is probably in all the peripheries around it. But here's a really interesting thing. If you cover this whole cell in light, you cover the whole dang thing, what happens? You want to guess? Which word does the brain understand now? More, less, or the same? If you have the entire thing covered, it's going to be the same, where basically these two are going to cancel each other out. The inhibitory surrounding parts are going to cancel out the excitatory center parts. But we'll talk more about that in chapter 3. All right. And that's it. Thank you so much for tuning in. Hey, we kept it to an hour. If you have any questions, let me know. Otherwise, happy studies. Have a great day, etc., etc.